Hello students, welcome back to the channel. In this video, we are going to study about quorum sensing. Now see, in the early days, bacteria were considered non-social organisms. Means they are living individual lifestyle with little interaction. But recent research have shown that many strains of bacteria, they communicate with each other by emitting or by identifying or responding to the chemicals. Ligands are the any chemicals or any molecules. And one important method which is studied in the bacteria to communicate between the two bacteria was quorum sensing. Clear, this was the one. It actually depends on the bacteria is producing one signal molecules which is going out from the bacteria. Now, bacteria will communicate to another bacteria through this chemical. It allows the bacteria population to communicate with each other so that they can coordinate their behavior after the particular population size is achieved. Okay, and it is useful for variety of functions in bacteria for the production of example, for the production of bioluminescence, for sporulation, for virulency means pathogenicity, or biofilm formation. So the chemical which they release is known as autoinducer. So when the bacteria population is low, this concentration of autoinducer in the surrounding will be low. Slowly, when bacteria population is increasing, concentration will also increase, and it is now becoming as a positive feedback to increase more and more production of this chemical. If you see the biofilm formation is nothing, it is a aggregation of bacterial cell attached onto the surface. And what they will do, they will generally derive the nutrition from the surface on which they are. You have seen the biofilm generally on your teeth early morning when you are doing the brushing. That time actually you are disturbing that biofilm. Is it that is why we say morning teeth is brushing is required to disturb the biofilm which bacteria has made. Now next, understand bioluminescence. How? Very good example of quorum sensing is by bioluminescence. We found one marine bacteria is there, that is Vibrio fischeri, which produce one enzyme that is known as luciferase. This enzyme is the one which can help in producing the proteins so that emit light can be there. So they will produce this enzyme when they will reach a critical cell population density. And this population density they have to achieve in the light organ of the host. And generally their host is squid. If you see both relationship is a mutual is why? Because bioluminescence, bacteria is getting benefit because bacteria is getting a place to live and it is getting nutrient. Squid is also benefit because it is actually the light which is emitted from the bacteria. It is used as a camouflage, or you can say it attracts prey. Now, how this interaction is working? See, when the bacteria is living inside the light organ of its host, then during its reproductive cycle, they will produce autoinducer molecule, which will pass out from the cell membrane into the surroundings. Because bacteria is reproducing, obviously the number of bacteria cell will increase. Slowly, what will happen? Concentration of autoinducer molecule will also increase. Now, when the number of autoinducer molecule have reached to the threshold level, they will come again into the bacterial cell. See the diagram, autoinducer cells are coming back again into the bacterial cell and then they will bind to the protein that is LuxR. This is an inactive LuxR to which they are binding and making it activated. This activated LuxR will now bind to the DNA and will start production of the protein, means which is working as an enzyme that is known as luciferase, which is responsible for bioluminescence. I hope so. This video might be helpful for you. Thanks for watching.